It is now 12:15 and I'm going to start. Good afternoon, everybody. We are very pleased to be hosting this joint endeavor between the Juvenile Diabetes Research Foundation and Health Canada, and we hope to learn quite a bit during today. The advances in diabetes have been very exciting from its very early days until today. And while we still have a long way to cover, I think we have made enormous strides as it is. I want to welcome you all to this afternoon session, which will deal with immunotherapy and biomarkers of diabetes. And we have 4 very distinguished speakers. We welcome your questions in English or in French. You may either ask them verbally at the end of the 4 presentations, or you may put them in the chat and they shall be monitored and they'll be prioritized. There will be, you also saw one poll, and there may be other polls that may be coming, so please pay attention, and we would like you very much to answer those. Comment as you fit on, I may or may not comment on the poll results, you can and it is my pleasure then distinctly to introduce our first speaker, which is Dr. Diane Weret, who is a pediatric endocrinologist at the Hospital for Sick Children, a professor at the University of Toronto, and Center Director of the Canadian site of Type 1 Diabetes TrialNet. She will provide an update on teplizumab and other strategies being explored to prevent Type 1 diabetes. And Dr. Weret, the floor is yours.
Thank you. Let me just get set up to do the screen share. And 2 seconds. Okay, so thank you very much for the opportunity to speak about the research that's being done in order to prevent type 1 diabetes. I'm going to highlight the teplizumab study, but also some of the general issues, and I have no conflicts of interest just to declare. So as a quick overview, I'm going to first review some of the key concepts in the design of trials for the prevention of type 1 diabetes. I'm going to highlight the results of one particular trial of teplizumab, and then also talk about what's going on in the landscape around the world in recent trials. And then finally, to highlight some of the challenges we face and some of the future directions that we see coming in the next few years. So as a quick overview, the foundation of the research into trying to prevent type 1 diabetes is really this model of the pathogenesis of diabetes that began to be developed in the 1980s. It's been long recognized that there is genetic risk of type 1 diabetes, largely recognized because type 1 diabetes is more common in family members of those living with diabetes than in the general population. We then know that a small segment of those at genetic risk will have activation of their immune system. T cells or T lymphocytes have been recognized to be a key initiator, but the full, all parts of the immune system are important. How that immune activation, which is really silent, is detected in both the clinical and research spheres is through the measurement of diabetes-related antibodies. And typically initially this is detected as the presence of a single autoantibody, but only about 20% of those who initially have a single autoantibody then actually have developed a more broad-based immune response that spreads to other diabetes autoantigens, and then there are recognized to be multiple autoantibodies and really a widespread immune response that then leads to the destruction of beta cells and the eventual diagnosis of type 1 diabetes. The very high-risk nature of having multiple autoantibodies had been recognized for many years, but much of the data that had been published really focused on 5-year risks that were typically described in the 30 to 50% range. About 7 years ago, this publication that brought together cohort studies from Germany, Finland, and Colorado, where infants who were at risk of diabetes, largely because of having a family member, were followed from birth. And this figure denotes the follow-up from the time they were found to have multiple diabetes autoantibodies at time 0, and what happened in terms of the probability of progression to diabetes over time. So the 5-year data was confirmed that had been published over many, many years, that this approach is 50%. But what this publication did was really extend our knowledge of the long-term risk of diabetes in those with multiple antibodies, and showed that by 10 years, the risk is at 70%, and by 15 years, the risk is at over 80%. And some smaller publications have continued to really indicate that the risk continues, and that the ultimate risk of type 1 diabetes in those who have multiple diabetes-related antibodies likely approaches 100%. So this growing knowledge from these studies from around the world really led about 5 years ago to a consensus conference and a consensus statement really trying to better define the stages that lead to the development of type 1 diabetes. And I won't reiterate the early stages that I already talked about in terms of genetic risk, though just to indicate this is largely based in the HLA complex, which accounts for about 40% of the risk, but there are multiple genes where polymorphisms are associated with increased risk. The immune activation is a subject of very intense investigation that I don't have time to go into at all today. And as I mentioned, there is the first line is often the development of single autoantibodies. So really, once someone reaches the development of 2 or more autoantibodies, this has now been defined as stage 1 of type 1 diabetes, and it is characterized by normal glucose tolerance in a research setting done with glucose tolerance tests. And so this is completely silent. Stage 2 then, with further progression of damage to beta cells and loss of insulin secretion, is defined by the continuation of the autoimmunity, but also early abnormalities of glucose metabolism, and typically found through glucose tolerance testing that is abnormal, but still silent. And then finally, the clinical diagnosis of diabetes is made after further loss of beta cells. Though at this stage of diabetes, there is a typically quite significant amount of residual insulin secretion left. And finally, after a period of for some months, for others many years, there is eventual loss of insulin secretion in what is now described as stage 4. And so this concept of staging really highlights that type 1 diabetes really begins here at stage 1, even though it is an asymptomatic stage, and which is in contrast to what we had always talked about, which is diabetes begins at the time of clinical diagnosis at stage 4. And I think one of the other really important paradigms that's come from the concept of staging is really thinking about the concept of treating beta cell autoimmunity, much as we treat other silent conditions that are associated with significant clinical outcomes. I think common ones we can think of are treating hypertension or treating elevated cholesterol in order to prevent cardiovascular disease. And we certainly accept that, and it is the paradigm. And we know that those at stage 1 and 2 have a 10 to 12% risk per year of progressing to type 1 diabetes. So that we hope that similarly in the future, treating early stage diabetes could potentially become as commonplace as taking a statin or an antihypertensive in order to prevent strokes and heart attacks. The field of intervening to try to prevent beta cell loss has been around for a long time. And there were early studies, in fact, one that was mentioned by the introductory speaker today, of the Canadian-European cyclosporin trial that happened in the 80s. Early studies were focused largely at the time of diagnosis. That's when patients are easy to find and eager for treatment that might improve or reverse diabetes. And the early studies showed some promising benefit, but not any sustained benefit, and often concern about side effects. In more recent years, with the development of multiple immunomodulatory agents that have typically come from the worlds of rheumatoid arthritis, inflammatory bowel disease, and other autoimmune conditions, it's been recognized that many immune interventions are able to help slow the loss of insulin secretion that happens after the time of diagnosis. And I'm going to highlight a couple of these, but just to highlight that there have been multiple drugs that have had an effect at this stage of diabetes. To go at the very opposite end, there have been some studies done in those at high genetic risk, typically newborns, aiming at trying to prevent this immune activation from happening with the concept of trying to remove potential agents that may target immune activation. Canadian centers participated in the trial to with avoidance of cow's milk protein. Gluten study happened in Germany, and unfortunately, neither of these interventions had any effect on the development of autoantibodies as an indicator of an immune response or of diabetes. Another large area of intervention has been trying to prevent progression from stage 1 or 2 to actually moving to diabetes. There were some early studies of nicotinamide done through a Canadian and European effort that did not meet its outcome. And there have been a number of studies using different forms of insulin in order to try to induce immune tolerance to insulin. And again, no big home runs there. There are 2 ongoing TrialNet studies, one of abatacept, a T-cell modulator, and one of hydroxychloroquine, where we will have results in the coming years. And then finally, to talk about teplizumab, this drug, which is a modified anti-CD3 antibody that making it less depleting of T lymphocytes, had shown good promise in the new onset world, and so the drug was used in those at risk, and I'll highlight some of the information about that study. How are these studies done? So typically, when we think of the very early stage studies, it's really aiming to prevent the development of autoantibodies or type 1 diabetes. And I want to highlight the duration here because these are long studies, so it takes a long time to both identify an adequate number of people who are at risk of diabetes, and then the outcomes take a long time to develop, as I showed with the graph looking at risk over 5, 10, and 15 years. When studies are done at stage 1 or 2, it's usually with the outcome of prevention of progression from stage 1 to stage 2 or stage 2 to diabetes. These again are long studies, not quite as long because the outcomes come a little more quickly. But again, finding those who fit these particular stages is like looking for a needle in a haystack. So that takes time, as does the progression from one stage to the next. So these are studies in which a large investment takes place in order for them to happen. Studies of recent onset diabetes, on the other hand, can be done much more quickly. The participants are much easier to find, and outcomes can be seen at 1 year with good follow-up to 2 years. The outcome that has really risen to the top is looking at the ability of the body to still produce insulin as measured by the marker of endogenous insulin secretion, C-peptide. And this is chosen as the primary outcome because it is very difficult in the first year of diabetes to really see differences in other typical clinical outcomes in diabetes research like A1C or hypoglycemia, because A1Cs tend to be very low in the first year after diagnosis, and hypoglycemia is relatively rare. So then to think about how these trials are performed, then you really have to think about how do you identify
2 minutes left.
thank you, identify at-risk populations. So that's done by in relatives by measuring autoantibodies, and they can be further stratified by other markers. Trials have also been done using those who aren't relatives, and these have largely been done using genetic risk scores or gene chips that include a high-risk HLA and the addition more recently of non-HLA polymorphisms, or in the measurement of multiple autoantibodies. And there is a large effort going on in Europe right now in order to recruit family member, or to recruit the general population through having genetic screening and enroll in a trial of oral insulin. And a similar trial was done in Germany using primary care visits for young children and identifying in that population about 0.3% and in the other up to 1%. Screening has very recently moved outside the clinical arena in that the JDRF in the US has very recently launched an effort to provide access to testing to anyone who wants it. One can go to the JDRF website and order a test, do a capillary sample at home. It's run through a lab that tests the four most common autoantibodies, and then a result is given. The hope is that this will help to prevent morbidity at the time of diagnosis, either through very prominent prolonged symptoms, and importantly, to prevent the development of diabetic ketoacidosis. And this will also hopefully identify additional people who may be interested in participating in trials of prevention. So I'm now going to turn to talking about the teplizumab study that I mentioned.
I'm afraid that you're pretty much out of time.
Sorry, I was told I had 18 minutes, and I've
got how much? Okay,
14. So I've got
okay, go ahead.
So this is a multi-center placebo-controlled trial aiming to delay or prevent diabetes with the primary outcome of the comparison of time to the diagnosis of diabetes. It was a small early phase 2 study in this population, done largely in people under 18. They had more than 2 or more diabetes antibodies and abnormal glucose tolerance. The drug is given only once in a 14-day course at the beginning, and then people were followed with a median follow-up of 2 years. As expected, over half developed type 1 diabetes, and the duration of the study was almost 8 years. This result shows the placebo group in red and the treated group in blue, and highlights the reduction in the hazard ratio in the treated group with a median delay of the onset of diabetes of 2 years. And you can see the very prominent effect of the drug over the first 12 months of therapy when we know it is most active, and then eventual development of diabetes that begins later on in the treated group. So what's happening with this drug? It has now been granted particular designation by both the FDA and EMA in terms of a breakthrough type therapy. An application has been for indication for delay or prevention has been submitted to the FDA and will be reviewed later this year. And there is also an ongoing multi-center phase 3 trial in recent onset children with type 1 diabetes, as they have shown the best efficacy in other studies. So this drug is making its way, and hopefully will be one that we will see exciting results. There's been also a recent new study of another immune intervention with an anti-TNF therapy that, again, in recent onset young people under 21 has shown preservation of C-peptide, and also importantly a reduced use of insulin despite similar hemoglobin A1Cs, again indicating that this is that type 1 diabetes in its early phases is responsive to many immune interventions. And this shows the results of the golimumab study, showing the typical loss of insulin secretion that happens over the first year in the placebo group in blue, with a much reduced loss in the treated group in brown. So just to summarize, the challenges that the field face. Clearly, as I've highlighted, finding at-risk populations is a challenge. The studies are long and therefore require retention, and this is particularly challenging in those who don't have a family member with diabetes. We would benefit hugely from the identification of biomarkers of disease progression and of efficacy of treatments that would help to facilitate pilot studies, shorter trial design, and perhaps allow alternate designs. And clearly, the teplizumab study is only the very beginning, and we need therapeutic approaches with much longer-term efficacy and, of course, importantly, long-term safety. So what's happening currently in 2021? There are a number of new trials that are ready to start and are actually on hold because of COVID, because of the concern of immunomodulation. So hopefully those will launch later this year as the vaccines become widespread and hopefully COVID numbers go down. Much work is being done on improving methods for identifying populations at risk, both by improving antibody assays and improving genetic risk scores and methods to do them. There's been some very exciting work around collaboration among the European, North American, Australian networks. And there are a couple of trials that will report out this year with both at new onset and then the TrialNet study of abatacept. So with that, I'll stop. And just as Norm Rosenblum did earlier, I'm going to highlight another important diabetes conference that's happening this year, the Insulin 100 conference sponsored by the University of Toronto, which is already available online, and the big discussions happen in April. So thank you.
Thank you very much, Dr. Weret. And now our next speaker is Dr. Bruce Verschaer, who is a professor at the University of British Columbia and is the head of the Diabetes Research Program at the BC Children's Hospital Research Institute. He will discuss moving towards better biomarkers for type 1 diabetes risk, onset, and progression.
Well, thank you very much. Yes, thanks very much, Dr. Klein. As she's just indicated, I'll be talking about biomarkers in type 1 diabetes and how they pertain to our ability to predict disease and to track the progression of the disease in humans and in response to clinical trial. And I think this will dovetail nicely on the presentation that Dr. Weret just gave. I have nothing of note to disclose. These are some advisory panels and advisor roles, but they are not of relevance to today's presentation. And I'll also start with a really brief outline. I'm going to introduce the concept of a biomarker, and in particular what biomarkers are used presently in type 1 diabetes and their value and limitations. And then I'd like to move into some new areas where we think the future of biomarkers will be in type 1 diabetes, particularly with regard to assessing immune cells and beta cell biomarkers, including some of our own research, and then we'll summarize. So let me start by defining a biomarker. The term is a portmanteau of the words biological and marker, and you can see the definition described here, but essentially they're measures of physiological processes or pathophysiological processes, and they can be used to predict disease, to assess disease progression, and to track the response to therapy, and they can be used in diagnosis as well. And typically they will come from a few drops of blood or a tissue sample, some DNA, and they come in several different types. So there are predictive biomarkers. Can we predict who will get disease and when will that occur? And you heard Dr. Weret talking about autoantibodies, so those are a good example in type 1 diabetes, and I'll talk a bit more about that in a moment. There are risk biomarkers, so we inherit genes that give us genetic risk of disease. There are prognostic biomarkers that enable us to assess how a disease will progress once you have it and in response to therapies. And there are diagnostic biomarkers that tell us whether we have disease or not and what type. And in the case of diabetes, this is important because it's a heterogeneous disease. So let's talk about these biomarkers currently in use in type 1 diabetes. And obviously, people on this presentation are likely familiar with the common ones, which include blood glucose, measured by a glucometer or continual glucose monitoring, hemoglobin A1C, and loss of C-peptide. And these can be done just from a few drops of blood. There are risk biomarkers. Dr. Ware touched on these. We inherit risk of type 1 diabetes. It has a genetic component to it as well as an environmental component, particularly the HLA genes involved in immune responses. And there's a lot of research going on now in the calculation or determination of a genetic risk score, which gives you a prediction of your genetic risk of developing disease. Predictive biomarkers are gaining steam in research, and autoantibodies, as Dr. Ware just pointed out, are most commonly used in clinical trials and are good predictors of disease, but have some limitations, as she alluded to. And there are prognostic biomarkers. Again, these are the ones that allow us to assess how disease might progress once it's been diagnosed. Autoantibodies have some value here, but particularly in use in clinical trial is the use of C-peptide to understand how beta cell function is progressing, if it's declining as the disease progresses. And one could put autoantibodies up here in diagnostic biomarkers as well, because it's used sometimes to definitively diagnose type 1 or autoimmune diabetes. So there are a number of biomarkers, but as you can see here, the primary ones in use in clinical trials and clinically are C-peptide measurements and autoantibodies. And these are very valuable and critical in clinical trials, but I believe we need more biomarkers that can give us better insight into disease pathogenesis, and I think have real potential to improve type 1 diabetes prediction, as well as assess the response to clinical trials and immune therapies. So Dr. Ware showed this staging diagram, how many of us look at the progression of type 1 diabetes. And I won't get it into too much detail here because she's already presented it, but I want to put it into the context of where biomarkers will have value. So we inherit genetic risk, and there's immune triggers and immune response that leads to a progressive loss of beta cell function. And trials can be given depending on the drug or the trial at various stages over the course of disease. Right now, the most common measure, as was alluded to in the last presentation, is C-peptide, the response to a meal or a stimulation. And that's really the best measure we have, and how C-peptide is lost as the disease progresses. It goes down, and can immune therapy slow that loss? But we're very interested in new markers of biomarkers that can assess the amount of beta cells we have left, whether they're living or dying, and how well they are functioning. And I'm going to talk more about those in the coming slides. And there's also measures of autoimmunity, the T cells that are attacking the beta cells, what their function and phenotype is. And I'll talk more about that as well, because these are really active areas of research. And I would argue that they're going to have great value, and they already do, in ongoing clinical trials across these stages in assessing the response to therapy, as well as predicting and potentially stratifying who should go into clinical trials. So I'll talk first briefly about some of the methods for assessing T cell biomarkers. So type 1 diabetes is an immune disease, an autoimmune disease in which T cells attack the beta cells. And we now have many ways in the lab in which we can assess the way these T cells attack beta cells. And these typically fall into antigen-specific approaches, those that are T cells that are targeting a specific protein or epitope on the beta cell surface, as well as those that are agnostic to the beta cell antigen. And they largely fall into 4 categories. And without getting into too much detail, they can detect the way T cells respond to seeing beta cell antigen by increasing their proliferation or producing toxic cytokines that can induce beta cell dysfunction. There's very cool technology using tetramers, which allow us with fluorescent probes to actually detect specific T cell receptors that detect beta cell antigens. And Dr. Santa Maria, who's following me, may be presenting some data on this. And there are also cytometric assays, particularly using flow cytometry, that allows us to count and phenotype T cells, single cells among millions, using fluorescent probes. And then emerging is the field of transcriptomics, which allows us, even at a single cell level, to look at changes in gene expression. So these technologies are quite game-changing. They have many benefits. They provide deep insight into T cell phenotype, the number and function, as well as into how they are responding to immune therapy. And an example is in the clinical trial that Dr. Ware talked about, teplizumab, the mechanism by which that drug is acting. We've gained insight into that from some of these T cell assays in these patients. But these, as you can imagine from this diagram, they're expensive, they're difficult, and they're not really used widely. They're used in the lab and in clinical trials because it's difficult to standardize these across many different laboratories. And I'll refer you to this really excellent review from Simi Ahmed, from which this diagram came. She's with the JDRF in New York. So let me talk about a couple of specific T cell assays, and then I'll move into the beta cell biomarkers. And this is work done by my colleague, Megan Levings at BC Children's Hospital in Vancouver. And she identified a gene expression signature in a particular type of T cell, a T regulatory cell, or a Treg as it's commonly called. And these cells are known to check autoimmunity and that they may be functional or decreased in number in type 1 diabetes. And these are the red cells here. And you can see as the disease progresses, they become lower in number and probably lower in function. And what Megan and her colleagues identified, and this work was led by a fellow, a JDRF-funded fellow in her lab, Ann Pestnacker, that there is a gene expression pattern in people with diabetes, whether new onset or if they've had established disease. And using this gene expression pattern in these T regulatory cells, she thinks that she may be able to better predict disease as well as track the response to therapy. And she has some data to suggest that that is possible in response to an immune trial, an immune therapy trial that's going on in Vancouver now. Let me talk now about beta cell biomarkers in type 1 diabetes. And so the insulin-producing beta cell is the target of the immune system in type 1 diabetes, and they're lost or dysfunctional in this disease, and you get loss of insulin secretion. And like other secretory cells in the body, beta cells release into the blood a lot of proteins and DNA and things that can be measured. So one, of course, is insulin, as well as other forms of insulin and other hormones that are made by the beta cell that we can detect using assays. But they also release pieces of the cell that can contain pieces of DNA and other proteins. And new technologies allow us to detect these in the blood. And these can have value in assessing beta cell death if they're dying, or if they're dysfunctional or functional, both in people with type 1 diabetes and potentially in at-risk individuals for predicting disease. So let's talk about a couple of these beta cell biomarkers. One really interesting area is this new predictor of beta cell death. So we think that that's an important event in type 1 diabetes. The immune system is killing beta cells, and dying beta cells release bits of DNA into the blood. And there are markers of beta cell DNA. Basically, a section of the insulin gene has this mark or doesn't have this mark on it called methylation. And new technologies called epigenomics allows us to detect these circulating bits of DNA. So it's a really amazing technology because they're circulating in tiny, tiny amounts, but genomics technologies allow us to measure them. And this was work pioneered by Kevin Harold at Yale a few years ago. And he was able to show in people at high risk for type 1 diabetes, that is, they had multiple autoantibodies as well as some dysglycemia, they had high levels of this unmethylated insulin DNA in their blood compared to non-diabetic control individuals. So it's quite a remarkable result, but you can see the spread is very wide, and lots of the high-risk individuals had so-called normal levels of this unmethylated DNA. So there's lots of work to be done here, but this is a promising biomarker, and the specificity and sensitivity of this method is still being investigated. There are issues with it, but the potential to be able to track beta cell death is quite interesting and promising, and other DNAs and microRNAs are being used and investigated for this purpose. I want to talk about other aspects of beta cells as biomarkers, and particularly beta cell function. And this is work I actually just presented yesterday at the NPOD meeting, a diabetes meeting that's going on, where scientists are presenting work understanding beta cell death and dysfunction in type 1 diabetes. And what we've learned from this important repository biobank of pancreatic tissue is that beta cells are not completely destroyed in type 1 diabetes. So if, we've been taught that, and it's in the textbooks, but we now know from these autopsy samples that, and here you see a number of insulin stains under the microscope for in pancreas sections from people who had type 1 diabetes at the time of death. And you see a fair amount of insulin producing beta cells. And so beta cells are still present in type 1 diabetes, and they're not completely destroyed. And this has a number of implications. I should say also that it's highly variable among patients and even within the same pancreas. But it implies that there are functional aspects to type 1 diabetes, that the beta cells are there, not totally lost, but also are dysfunctional. That these are a target for regeneration. Could we wake these cells up or make them replicate to increase the number of beta cells? And importantly, and where I want to go now, is that can they be used as biomarkers? Can we measure markers, proteins and things that are released from these residual beta cells? So I'll talk about two, and these are prohormones. And as background, I want to tell you that insulin that you see here is made from a larger molecule called proinsulin, and it's processed in the beta cell by a series of enzymatic steps that are very specific and result in one molecule of insulin and one molecule of C-peptide being made. And that's why we use C-peptide as a surrogate measure for insulin in people with type 1 diabetes who are receiving exogenous insulin. C-peptide is made in what we call equimolar amounts, equal amounts to insulin by this process. And we've known from other studies and for a number of years now that proinsulin actually is a good biomarker in type 2 diabetes of beta cell function. So proinsulin levels, the amount of proinsulin relative to the amount of processed and mature and biologically active insulin, is quite markedly elevated in people with type 2 diabetes. So it's a good biomarker of beta cell dysfunction in type 2 diabetes, as well as having some value in disease prediction. Recent studies now have shown that these residual beta cells in humans with type 1 diabetes are persistently secreting proinsulin. This is a really beautiful study done by a group in Indiana, just published a year or two ago. And across a broad spectrum of patients with type 1 diabetes, of varying disease duration, even more than 10 years or relatively newly diagnosed, and of varying degrees of C-peptide, including those who have undetectable levels of C-peptide in their blood, you can still detect proinsulin in almost all of these individuals. This suggests that these residual beta cells in people with type 1 diabetes are not working well. They're not producing insulin and C-peptide from proinsulin, but they're sort of releasing proinsulin into the blood. And so this may be a biomarker of these residual beta cells. And it may even have value in predicting disease. So this is again from the Indiana group, and they measured the ratio of proinsulin to the amount of mature C-peptide in the blood in autoantibody positive people who either did not develop the disease or who ended up progressing to developing type 1 diabetes. And the first thing you'll note is that there's a great spread and overlap here. So its usefulness in predicting disease still needs further investigation. But what you can see is that the amount of proinsulin relative to mature C-peptide in people who subsequently went on to develop diabetes is elevated. So it may have value in predicting type 1 diabetes. And I'll close by presenting a little bit of our own work. So we've been interested in another peptide hormone made in the beta cell called IPP. It's co-secreted along with insulin, and it's also processed from a larger precursor form that we call pro-IPP. And a couple of years ago, we developed an assay, an immunoassay that allows us to measure one of these circulating forms of this incompletely processed version of IPP that we call pro-IPP 1 to 48. And using this assay, we made what I think is an interesting finding. So in kids with type 1 diabetes, these were kids in a BC Children's Hospital cohort, they had very low levels of the mature form of the peptide in their blood, very similar to what we would see with a C-peptide in these patients. But when we measured this pro-IPP form, very much like what the Indiana group showed with proinsulin, we found that the pro-IPP levels were elevated, almost as high as healthy non-diabetic individuals. And so when we calculated that ratio, the ratio of the incompletely processed form to the mature form, it's markedly elevated in children with type 1 diabetes compared to non-diabetic individuals. And so we think that pro-IPP could be a biomarker, and we think it could go hand in hand and be complementary to proinsulin. And you can see we have replicated this in a cohort from with the Indiana group, where they measured proinsulin to C-peptide, and here you see the elevation in people with type 1 diabetes. We similarly saw increased in pro-IPP in the same patients, and there was a relationship in these patients so that those with high pro-IPP levels also had high proinsulin levels. So we
Dr. Bashir, you have a couple of minutes left, that's all.
Yes, I'm on my last slide, thank you. So let me close then by summarizing the field of biomarkers in type 1 diabetes and where we think this will go. So we see that in a child or even adult with type 1 diabetes, someone may be entering a clinical trial or at risk of developing disease, that just by collecting a few drops of blood, we're going to be able to measure many things. Some we already do, such as islet autoantibodies and C-peptide, but more and more we're able to identify risk genes and calculate their genetic risk score. I mentioned the assays for T cells that attack the beta cells, phenotyping them and quantifying them, as well as new measures of beta cell death and function, including our own work in prohormones. And we can not only track them at diagnosis or even prior to disease, but over the course of clinical trials so that we can better understand how they are responding to therapy. There are also a number of biomarkers in the pipeline that are being investigated, including the microbiome, epigenetics. Can we image beta cells using PET or other techniques, MRI, and new beta cell biomarkers? And many groups, including our own, are trying to develop composite biomarkers. Can we combine these various measures to come up with unique measures that can predict diabetes? And we think where this will go is it will allow us to define disease heterogeneity, the different endotypes of disease, because diabetes is a heterogeneous disease. Can we better stratify patients for clinical trials and understand how they have responded to therapies? And importantly, can we improve the treatment of people with diabetes by understanding the type of diabetes they have, and some of these biomarkers, can we refine their treatment to make it more precise? And in the end, I think all of this will combine, and these biomarkers will give us new insight into the pathogenesis of type 1 diabetes. So I'll stop there, and I want to thank the funders, particularly of this biomarker work, JDRF and the CIHR, as well as a number of trainees and collaborators who have helped with this work over the years. Thank you.
Thank you. Thank you, Dr. Bashir. That was excellent. Okay. So we have an overwhelming majority of attendees from Ontario. Do you want to let's see, you can go to the next set of questions.
Was that for me? You want me to answer that question?
No. I was talking to Andrew.
Okay.
We're going to put up a couple of questions, polls.
That was it, Agnes, or that's it. We're good to go to the next speaker, Agnes.
Okay, we're good to go to the next speaker. All right, so the next speaker will be Dr. Perez Santamaria. Dr. Santamaria is a professor at the University of Calgary, and his talk is entitled Peptide MHC-based Nanomedicines to Treat Autoimmunity. And of course, that's got a really important element for today.
Hello, everyone. So let me share the screen 1 second. Okay. Alrighty. Can you see the screen? Yep.
Yes, perfect.
Okay, perfect. Okay, well, thank you, Dr. Klein, for your introduction, and to JDF Canada for putting as well as the Health Canada for putting these series together. It's been fun. So I have a conflict declaration to make, so I'm just going to go back. This thing is moving by itself. Who has control? I mean, so can I get control? Okay, sorry. This thing was moving by itself. So I'll be talking about peptide measures to combat already complex-based nanomedicines to treat autoimmunity. I have a conflict declaration to make, which is I'm citing founder of Biomarkers Therapeutics, which is related to the talk that I'll be giving. So the agenda is I'll be discussing, the basic fundamental challenges, therapeutic approaches that are currently under consideration for the treatment of not just type 1 diabetes, but also other autoimmune diseases, and the pros and cons of each of these approaches, general approaches. I'll then be summarizing and describing what peptide MHC-based nanomedicines are. I'll talk briefly about mechanisms of action, and then I'll be disclosing or describing how these drugs can be harnessed for the treatment of various several different autoimmune diseases, at least in animal models of human diseases. Next slide. In autoimmune diseases like in type 1 diabetes, the most fundamental problem, with regards to how to approach the treatment of these diseases, is that they are antigenically very complex. What this means is that the immune system does not exclusively target one protein or one antigen in a given autoimmune disease, but targets many different proteins and pieces of those proteins simultaneously in the course of the progression of that disease. And that makes the use of or the development of approaches to selectively blunt the autoimmune response, again, this is moving by itself, the autoimmune response very challenging, in an antigen-specific way or in a disease-specific way, very challenging. In the case of diabetes, you have this is a cartoon that basically summarizes what happens during the progression, the onset and progression of type 1 diabetes. So this is a cartoon that shows a pancreatic islet. We have these green cells, which are the cells that make insulin, so insulin-producing pancreatic beta cells. In individuals who are genetically predisposed to developing type 1 diabetes, we think that an insult, specific insults to these pancreatic beta cells result in the shedding of proteins or antigens, so-called antigens, from the beta cell into the milieu, into the bloodstream, as well as the lymph. Then these proteins are then captured by what the so-called professional antigen-presenting cells of the immune system, which capture these proteins, chop them into small pieces, and then engage into handshakes with autoreactive T lymphocytes, which we all have in our peripheral immune repertoire. And then these cells become activated, and
NDG. What is it?
Okay, and this light disappears. I don't know what's happening. So the cells become activated, and then they migrate through the bloodstream back into pancreatic islets, and eventually start to kill pancreatic beta cells. So you can see an islet here that has fewer green cells, so the beta cells have been killed, and now you have infiltration of the pancreatic islet by these lymphocytes or killer lymphocytes, which are engaging in destroying pancreatic beta cells. And so when that happens, there is additional release of antigens into the milieu, which really results in additional priming of autoreactive T cells, additional activation, additional generation of killer lymphocytes, which then keep on killing pancreatic beta cells. And that results in spreading of the autoimmune response to numerous, what we call beta cell autoantigens, or numerous beta cell proteins. Now, if we think about therapeutic modalities in autoimmunity, we can think about three general therapeutic modalities. The first one would be an immune reset, meaning using approaches that will induce systemic immunosuppression to a greater or lesser extent. That includes the use of cyclosporine, azathioprine, etc., as well as current biologics which cause partial immunosuppression. That means that the immune system is handicapped, it's impaired to some extent, and that in chronic diseases like type 1 diabetes or other chronic autoimmune disorders, that exposes the patient to the risk of infection and obviously the risk of cancer in the long run. Another potential therapeutic approach would be to try to delete each and all of the autoreactive T cell specificities that are involved in a given autoimmune disease. Unfortunately, because of the diversity and our incomplete knowledge of these antigenic repertoires, this is not a pharmacologically viable approach to the treatment of autoimmunity. It would take hundreds of drugs given simultaneously to treat a given autoimmune disease for a given patient, and obviously that's not workable. The last general bucket of therapeutic modalities in autoimmunity is approaches that might be able to induce what's called antigen-specific regulatory cells, meaning immune T cells that can suppress autoimmunity in a comprehensive way without suppressing normal immune responses. Unfortunately, this is easier said than done, as there are currently no approaches that can actually do that, certainly not in the clinic. So a few years back, we discovered in the act of doing curiosity-driven research, a new class of drugs, which we call them peptide major histocompatibility complex-based nanomedicines. It's a mouthful, I understand. So we call them Navasims. And they represent a new class of drugs that can actually induce what I just said in my previous slide, the induce disease-specific regulatory T cells, disease-specific regulatory T cells that can then suppress the autoimmune response in a comprehensive way without suppressing normal immune responses. So in this slide, I'm just showing a bit of a cartoon showing what these nanomedicines are, how they are made. So there is an iron oxide nanoparticle. I mean, one can use particles made of other materials, but we use iron oxide because it's biocompatible and biodegradable in biological, in vivo. So these nanoparticles, which are extremely tiny, are decorated with they are functionalized, not decorated, functionalized with molecules that can accept in a directional way what the effector molecule that is responsible for inducing the regulatory cells. And these are the major histocompatibility complex molecules, which we've learned how to express at high levels, purify relatively easily, and these molecules present a disease-relevant antigenic peptide or target at the very end, at the tip of the molecule. And then these molecules, multiple copies, are decorated or coated onto these nanoparticles. Now, what these nanoparticles do, just to give you a brief sense of how they work, is when they're injected systemically, that means intravenously, they find a needle in the haystack, meaning the autoreactive T lymphocytes, the killer T lymphocytes that happen to have an antigen receptor that is specific for what we decorated or coated on the nanoparticle. And then these cells attach to the nanoparticles, or the nanoparticles attach to these cells like Velcro, and they form these sort of clusters of antigen receptors on the surface of a lymphocyte. So this is an electron micrograph of a lymphocyte that is binding to these nanomedicines. There are multiple nanoparticles here that are attached. They aggregate and they form these clusters, and they are here magnified on this particular enhanced image, a large image, and then that induces massive activation of that lymphocyte, and that results in the reprogramming of the lymphocyte from being a killer into being a regulator, meaning a disease suppressor. And again, I don't want to delve into the detailed mechanisms of action, which are rather complex from the immunological point of view, but to summarize what, the general mechanism of how this works is you have, an antigen specific, or the diabetes causing white blood cell or T cell in your peripheral immune repertoire. There would be thousands of those, but the nanomedicine, you can use a nanomedicine that targets just the 1 out of the 1,000. That nanomedicine will find that one cell out of the 1,000, will bind to its surface with this receptor here, will induce massive signaling, and then it will reprogram that killer into a cell that retains the antigenic specificity, meaning the diabetes specificity, but now instead of wanting to kill pancreatic beta cells, wants to suppress inflammation of the pancreatic islets and suppress disease. And then this reprogrammed T regulatory cell expands massively to the point that you can measure its presence in peripheral blood as well as other lymphoid organs. And then these cells, which function as a biomarker of therapeutic activity and pharmacodynamic activity, they suppress disease through a number of mechanisms that I'm not going to elaborate in here. And they do so in a very disease-specific manner without compromising at all, as far as we can tell, the ability of the immune system to mount immune responses to viral infections, or to mount normal immune responses to cancer cells. Just a few examples of how these drugs work and to what extent they work in animal models. First, of type 1 diabetes. So this is a graph that shows on the vertical axis the percentage of mice that have reverted to normal glycemia, meaning normal blood sugar, in response to treatment. And then in the horizontal axis, you have the weeks after initiation of treatment. Time 0 is the time when the animals have developed high blood sugar, meaning they're diabetic. These animals are spontaneously developing diabetes, so we start treating them at this point. And in red, in yellow, and blue are 3 different type 1 diabetes specific nanomedicines. And then in green and in black, you have negative control, meaning nanomedicines that should not do anything on the disease process. And you can see that in this particular set of experiments, between 90 and 100% of the mice that were hyperglycemic reverted to a stable normal glycemia in response to treatment, and that effect was a lasting effect as well. Now, these drugs, the beauty of these drugs is that they can be harnessed by making small modifications to treat other autoimmune diseases. So you can imagine this as if you had a screwdriver and you want to change the bead in the screwdriver. So here's the beads, and you would have one for type 1 diabetes, one for arthritis, one for multiple sclerosis, and so on and so forth. Sorry, this bead would be this little blue thing that sticks out of the nanoparticles. You have the nanoparticle in yellow, and you have the measure is the complex molecules, which would be pretty much shared from disease to disease. And then all you need to change is this little bit at the end, and that will change the specificity of the drug, meaning that you can have almost essentially the same drug with some modifications that will allow you to go from one disease to the other. This is just an example of how we went from diabetes to, in this case, a model of experimental arthritis. So here the animals are, induced to develop swelling of their joints, so develop arthritis, like rheumatoid arthritis. And then once the animals have overt joint swelling, we began then treating them with an avacin that was specific for rheumatoid arthritis in red, or with we left them untreated treated with control nanomedicines in blue, as well as another control nanomedicine in green, which actually is a nanomedicine that was designed to treat multiple sclerosis. And you can see how the red navacin, which is designed to treat arthritis, how it blunts the progression of joint swelling and eventually enables the restoration of normal joint structure. So it repairs, it allows the normal reparative processes to repair joint destruction occurring in response to the autoimmune disease. And then this is just another example, but for a different autoimmune disease. This is an autoimmune disease of the central nervous system called multiple sclerosis. This is an animal model in this particular cartoon of multiple sclerosis called experimental autoimmune encephalomyelitis. So the animals are induced with a disease. The animals develop paralysis, limb paralysis. So the animals are lying on their backs. Essentially, they cannot move their tail. They are mice. And in the vertical axis, we have the
Dr.
score,
Santa
the
Maria,
severity
you
of the
have
disease.
2 minutes left, please.
Yes, I'm almost done. Thank you. And so we start treating the animals when they reach the maximum disease scores. This thing moves on its own. And you can see that in blue, which is a negative control, if you treat them with control nanoparticles or you leave them untreated, the animals have a persistent state of paralysis. But if you treat them with a multiple sclerosis, this specific navacin at different doses, you can see how the animals quickly begin to regain motility, and eventually they look not completely normal, but almost normal to an experienced investigator. We also have evidence that the human disease-specific drugs for these diseases will operate in human peripheral blood T cells like they do in mice. So we've done these experiments in humanized mice. Humanized mice is a mouse that can be engrafted with peripheral blood and mononuclear cells from patients. And this is just an example. I mean, it's a flow cytometry staining profile. And I don't think for those of you who are not familiar with these cartoons, you're just going to have to take my word for it. But basically, this area where there is circled dots, these are the regulatory cells that have induced and expanded in response to the human navacins for type 1 diabetes. And you can see that these dots are significantly increased here in the treated mouse as compared to the untreated mouse on the right-hand side. So in summary, and that's my last slide, is peptide-based nanomedicines represent a new class of drugs that is capable of reversing immunologically complex autoimmune disorders, including murine type 1 diabetes, in a disease-specific manner without compromising normal immune responses. And these compounds find autoantigen-experienced effector cells, meaning cells that partake or participate in the disease process, with the defined transcriptional epigenetic program. We know now what these cells, the original cells, the source cells that are reprogrammed are. And we know that they are poised to become regulatory in response to these nanomedicines. So these nanomedicines engage these cells and reprogram them, redifferentiate them into disease-specific suppressor T cells that are responsible for orchestrating a very complex cascade of cellular events that suppresses local inflammation and the autoimmune disease. And this is just collaborators as well as trainees and associates who have worked with us on some of the data that are shown on these slides. So thank you very much for your attention.
Thank you very much. Thank you very much, Dr. Santa Maria. Our last speaker for this panel will be Dr. Simon Sauvé, my colleague, who is the Scientific Research Manager in the Center for Biologics Evaluation in Health Canada. And his talk is entitled Refuting Bad Science, the Insulin Content of Products Marketed in Canada and the Adaptability of Pharmacopoeial Methods for Novel Product Regulation.
Merci beaucoup, Agnès. Vous pouvez voir mes slides très bien?
Thank you very much, Agnes. You can see my slides very well. Yes, we can see them. Perfect. Thank you, Agnes, and thank you for the organizers from the GDRF for having allowed me to present today. And on top of that, presenting in French, that is a blessing. So thank you so much. So today my presentation will talk about a study that we did after publication that suggested that possibly there were problems with the content of insulin in the pharmacies in Canada. And so we wanted to make sure that it wasn't the case. And on top of that, we will take the chance to see how the approved methods are necessary to make sure that it's not the case and to show that products are regulated by Health Canada. So I wanted to say who we are first. Before yesterday, I was part of the Separation Sciences and Analytical Biotechnology Laboratory. So what we want to do is development and applications of analytical methods for biologics based on high-resolution separation techniques. So not forget anyone, the team that has worked on this project, some members of the laboratory, in particular Barry Lorbetsky and Stuart Bigelow, and also Lisa Walrond and for the spectrometry. And we see it's funny that the Banting is the name of the center of research. We're talking today about insulin. So that's interesting. So I'm going to give you a little bit of background and regulatory context, but I won't take too long to do this. But even though that the Dr. Rosenblum really did a good work with this, but of course the diabetes is a health major problem in public health, and so the associated costs are
370 millions et plus de personnes affectées avec des coûts associés à 471 milliards de dollars
of $400 billion American. And the market is estimated as $42.9 billion in 2017, when we're saying that there's going to be a substantive increase in 2025. It's a big market and with a lot of products that can help to manage diabetes, as it was presented by some of the presenters today. So for me, to talk about the study that we did, we're going to talk a little bit about like a 101 insulin course. I'm happy because Bruce Verscher spoke about C-peptide as a biomarker. Today, I'd like to see the chain 1 and chain B here, you see. Insulin is a peptide hormone, and it's important for the insulin to be curved, to be effective, and to link the receptor to the insulin. So this is the pre-proinsulin form, and then there's a folding and disulfide bonds with proinsulin. So as Dr. Bruce mentioned as well, Verschere. And then we have the active form of insulin, which is composed of chain A and chain B. What's important to understand the next slides is to keep in mind that you have two forms of insulin, if you would like to see. You have the hexamer form on the left, which is the storage form where insulin is inactive. And to become active, it has to become monomer. So in the next slide, what's important here, as you have seen with Dr. Santamaria presented, how they could modify these molecules on a microscopic level for particular needs. And this is the same thing that we're doing here. In our case, the insulin is a hormone that is pharmacocinetic, and dynamic is short lasting. So how can we use insulin as a therapy to reduce the symptoms of those who suffer from diabetes? We started quickly work on this formula. So the different insulin products that are available, there are modifications that are made to allow to have a different different dynamics. So you have, for example, fast-acting insulin. So the stored form of insulin is hexamer, and the active is monomer. So if you want it faster, you want to destabilize the hexamer form and stabilize the monomer form. So the example that you're seeing here is you're destabilizing the hexamer form, so the distribution of insulin is much faster. And on the other side, if the distribution of the insulin is too fast, the effect will not last long enough, and then you'll have to inject yourself often. So patients use cocktails of insulin with fast-acting insulin and long-acting insulin. So in the opposite form. We engineered things to make sure that the insulin that was hexamer would be more stable, so it take longer to release the active insulin, and it would last longer. So here, what you have here, the insulin glargine, and we uniformed, and it stabilized the hexamer form of insulin, as we showed here. However, what we can do more, we can increase, do oligomers. So these are the engineered insulin. So here you have two examples here, insulin detamir and insulin degludec. So this is chain A and chain B of insulin was modified. So those are chemical modification. So it's to increase the oligomeric form of the insulin, so it increases the long-lasting effect of the insulin. So this is what we need to make sure to have a base level of insulin sufficient, basal amount, to lower the symptoms of diabetes. As I said in the beginning, the reason why we decided to do the study that I'm going to show you is that there were a paper that created a lot of noise in the media. It was a paper that affirmed more or less that there was a problem with the products, insulin products that existed in North America. And of course, this entered the media, and quickly it brought a lot of attention to these products. So quickly, the study presented is that they were using another method to evaluate and measure the concentration of insulin in the products that were manufactured and distributed in Canada, especially. Their observation was that none of the products had a sufficient content of insulin. So this is what you're seeing with a mass spectrometry. You see the concentration optimal should be between 95, about 95 units per milliliters, or 100 units per milliliters. As you see, none of the curves had these values, and you have the quantification on the bottom, and it's evident. So in some cases, it was less than 25% that was present, as the insulin was present in the samples. So the publication suggested that it was maybe a problem with the cold chain. So maybe the samples were not well kept in the cold, and there was a degradation. So consequently, that means that patients may have been receiving inadequate insulin. So of course, there was a big impact with patients that saw this. Did they say that they probably wondered, is there a problem with the medication that I'm taking? And of course, pharmacists asked the question, and they said, is that the case? And of course, other groups that support people that suffer with diabetes. So in response to this, we decided to do a very simple study. First of all, evaluate the claims of product instability in marketed and approved insulin products available to Canadian diabetes patients. And so we decided to use approved products. So of course, we wanted to validate and recognize high performance liquid chromatography methods. I'm going to use HPLC as the acronym to go faster. And we were using both the United States Pharmacopoeia, USP, and European Pharmacopoeia, EP monographs. So we wanted to validate and actually start again using the high performance liquid chromatography method, or HPLC methods, that were used in the previous study. So what we went to go buy every product that was possible for us to buy in pharmacies in the national capital. So we evaluated 24 different products. You have its list as well as a DIN. And then we went to the lab to see what happened. So I'm going to show you just a few results, lab results. And all the data that you're seeing is going to have to be represented in the same way. So this is based on the elution time on the bottom. So this is the concentration of the solution. So solutions of insulin medication is pretty simple and contains bacteriostatic agents like M-cresol. It could involve the protamine sulfate ingredient, which would increase the biodistribution length of insulin. And here you have the blue is insulin. So these curves are well adapted because you're actually separating the various ingredients. So you won't have interference of the other molecules that are in the sample. So then I'm going to go to the other part. I know that this is a very heavy slide, but we included all the products that we evaluated. And as you can see, the various forms of insulin has a different distribution rate. So since when we want to we work with the various insulin, as like we mentioned before, to either make it last longer or last faster or act faster. So if you want to favorize a hexamer form of insulin, longer it's going to last. So Lispro, which is considered as a fast acting than normal insulin. So you can see that its curve is a little bit earlier than insulin. So it's around the 18 minutes. And while basic insulin is around 19 minutes. So what's interesting is to see that in these cases, all the products could be well separated here. As you can see, you have Lispro here, glulicin here, aspart here, recombinant insulin here, and recombinant insulin that is cut with isophane or protein that comes from fish. But for those products that were engineered, the long-acting, the deglutec and determere. So what was interesting for us was to see how much insulin there were in those samples, to see if there was a problem. Was it true that we had less insulin than we were supposed to have? So Barry modified this method in the laboratory to create a gradient, so allow a longer elution. So more or less, you'll see a similar profile. So the elution level stays the same, but we see a gradient that reduces the affinity of insulin for the column where you see the separation. So we were able to elute degludec and the determere. So we were able to see that the quantity of insulin at, just looking at it quickly like that seemed equivalent. And the next slide, you see the quantification list of insulin for all the samples that were presented. And good news, there was no surprise. All the values were received confidence of 95 to 105%. So the quantification shows that there was a similar content for the other tested samples. So that's the relative quantification. So for the samples that we tested, there were no problems. So we expected that a little bit because there was no increase of claims of patients that had diabetes that affirmed that they had difficulty with their products or any problems related to the control of insulin. So for the last part of the presentation, we tried to reproduce the experience that was the test or the trial that happened in the report, or that was under study in the report. And we happy to see that the method that was used in that study was really developed to do a pharmacokinetic monitoring, sport anti-doping analysis, and forensic toxicology analysis. What's interesting is that these were all samples for insulin and plasma. So that's interesting because insulins suffer from a poor transfer into gas phase. So especially when we want to do mass spectrometry. So spectrometry, sorry. So there was non-specific binding, poor sensitivity, poor solubility, and fragmentation. So because insulin is difficult to analyze by these methods, mass spectrometry and liquid chromatography. So it was impossible to have a good reproduction of this test because this was a random actually there was a poor reproducibility due to non-specific binding and poor mass transfer. And we expected to have equivalent concentration, but the technique was not available, not good. And so we want to use another method, and it needs to be compared to approved method with many samples. So this is what we saw. There were no problems. The quantity was sufficient, but we just wanted to see in this case that it's not a method that's approved or used to determine the quantity of insulin in the products. But if we had to develop a method, we would do it certainly by comparing it to methods such as the interpreter did not hear what method. But I'm sorry for going very fast, but the message to retain is that the purchase product tested contained the claimed amount of insulin. The mass spectrometry analysis of insulin in solution is difficult. And what we wanted to do with this study is that we want to be careful when we're using unvalidated analytical methods when we're talking about the quantity of products. It's very important to use methods that are approved. And we understand that we want to develop new methods, especially with the new products that are coming. It will be necessary, but we still have to be careful. We can't jump to conclusions because it could have a very important incident.
La dernière chose qu'on veut, les patients perdent confiance dans la médicamentation qu'ils utilisent.
We don't want the patients to lose confidence with the medication, the pharmacists and their doctors. You know, in relation to trying to improve their well-being. So finally, Canadians should be confident in the improved and marketed insulin drugs products sold in Canadian pharmacies. So there's enough controls in place that the capacity and the safety of these products is good for patients. Thank you so much.
Thank you very much, Simon. Andrew, can you put up the polls quickly?
The polls have been disengaged, so they're not there anymore.
Okay, that's fine. Can you kind of let the video go? We need to go on to the panel questions and answers. I've been monitoring the chat. I haven't seen any questions really. However, you can raise your hand. You can raise your hand and ask the questions. I think this has been actually a very, very interesting panel. It sort of takes me back to the days that we were working with the cyclosporin, and my goodness, we've come a long way in immunology and the like. And I'm really looking forward to how this field is going to develop. And it's very encouraging to hear that the teplizumab has been actually to some extent successful. When do you think, or how do you think the process and the timeline for developing really biomarkers so that this can be implemented on a broader basis as a preventative, the way Dr. Barrett sort of put it? Can each of you make a comment to that purpose, for that purpose?
And you want to go first in order of?
Sure.
Okay. So your question is really about how biomarkers would be part of the implementation, or?
Exactly, and what is a sort of a time frame you would envisage that this could be made a reality?
So the biomarker piece, I think, is, as Bruce highlighted, there's some really promising things, but it's also a really challenging field. I think there are some emerging things with the teplizumab in terms of some of the molecular analysis, the flow cytometry and the other analyses that are done that I didn't even get to talk about in part of the trial. So there are some potential biomarkers, but as Bruce pointed out, things done by flow cytometry are labor intensive and expensive. So they're going to be, I think, at least from the ones I've seen, but he may know more. And I think it'll be very, in terms of the other part of it becoming, to see the light of day for patients is really how the regulators, FDA is the first regulator to look at this. So how are they going to look at a small trial of only 76 people and whether that will be enough to, give an indication, but we'll see. And there certainly is lots of data in new onset diabetes, but I'll stop there.
Yeah, maybe I can add on, Dr. Klein. So I agree with Diana, and I think what there has been in trials like the teplizumab trials is lots of ancillary lab studies that have gone on to try to understand. So people have accessed those samples. I know, for example, that the Indiana group is, wanting to look at beta cell hormones in those samples. Alice Long and others have been looking at the T cell phenotype, and I think have gained insight. And I think that's in the publications, and Paradigm might know better than me, but I think there's evidence for T cell exhaustion and these, and they go to sort of an anergic state. So we're learning some about the mechanism by which these work. And maybe just add that, because I was asked a sort of related question in a direct message to me, which I'll just raise, is someone asked, how close are we to sort of widely applying these biomarker technologies for disease prediction in the general population? And I'd say we're moving that way, but we're a ways away because it's just not cost effective, right? But as things like genome sequencing and genetic risk scores, these things become cheaper and more available, I think we'll move to a place where disease prediction, diabetes prediction, it's never going to be a perfect science, but will get better and cheaper and more broadly applied in the general population.
Dr. Santamaria, any comments from you?
No, not really. I mean, I think I agree. I concur with what the other speakers have stated. I mean, flow cytometry and the use of, for example, tetrameric reagents, which Dr. Brasher has alluded to, will be important for us, for our technology. But in terms of applying those tools to the to drugs like the teplizumab, which we don't really understand the mechanisms of action really, it's going to be very difficult. I think for those treatments, one will have to be a little bit more humble and not humble, but I mean more nimble. And focus on biomarkers that provide the essential information that you want to get from the trials, without having the potentially confounding effects of not really knowing, what association there is between the marker, the biomarker, or the pharmacodynamic activity of the drug vis-a-vis the therapeutic activity of the drug. These are completely different things, and one has to be careful as to how these biomarkers are used, but said that, I think that developing new biomarkers for not just diabetes, but for other autoimmune diseases, particularly to understand the pharmacodynamic effect of the drugs, whichever they are in patients, is going to be really, really important. And I think we need more biomarkers, better biomarkers, more specific, more sensitive. And I'm not saying anything new that nobody else knows, but that's a fact. But I wouldn't say that what's being done in research can be directly translatable today, in terms of biomarkers to general applicability because of the costs, complexity, and so on and so forth.
Thank you. Any questions from anybody? You can unmute yourself and ask the question directly.
There is a hand up.
Hi, yes, it's Angela Noble from Health Canada. Dr. Santamaria, fabulous talk, thank you. Do you envision that technology having to be developed to be patient-specific? Whereas if I was at risk of developing diabetes, you'd need a blood sample to figure out what autoantibodies I had? Or do you think we'd be able to move towards having just your nanoparticle that prevents diabetes in an at-risk population? I know you can't directly take research and put it into clinical practice. You already mentioned that, but I was just wondering what you think is the future for this technology.
Yeah, so thank you for the question. The drugs are not, the nanoparticles are not personalized medicine. I mean, that's important to distinguish. I mean, it doesn't, what I mean by that is that one does not need to, take the patient and screen the patient for a bunch of things and develop a drug, a prospective drug for that patient. So the drugs will be developed, are being developed. They will be, and there'll be like 2 up to 3 different drugs for the entire population of type 1 diabetic patients. We only need one piece of information that can be easily obtained from the patients, and then the drug will be taken from the shelf and then assigned to, one of the 2 or 3 drugs will be assigned to the patient as a function of that little piece of information that we need. But the drugs will be manufactured and be ready to be used and tested in this way. So it's not personalized medicine. Otherwise, if it was personalized medicine, it would not be pharmacologically viable, because these drugs they're not easy to manufacture. They're a new class of drugs. We basically had to blaze the trail and learn how to manufacture, scale up, and develop all the new knowledge to advance them towards the clinic. And we've overcome many obstacles along the way. And so, the drugs are not personalized medicine. Just a short summary to my answer, and we only need a little bit of information, very small piece of information that can be reliably and quickly obtained from the patient before the patient is assigned to the drug.
Thank you, Dr. Santamaria. Just one follow-up. It sounds like you may have a platform to attach all these different antigens. Just if there is a shift, I'm just thinking into the future, is there ability to adapt that if there was a shift in the type of antigens that were being targeted for certain autoimmune disease? Or is it so complicated that would be, oh, here we go back to the starting point?
No. I mean, it's a platform technology. I mean, that's one of the beauties of this approach is that all the know-how that we have developed in the development of one drug is applicable to and extendable to the development of any other drug for any other autoimmune disease that we might want to pursue. Of course, that for every disease, one has to do some legwork to decide, what the specifics of the drug will be. But it's a platform technology, so all the drugs from one disease to the next will be essentially identical.
Thank you very much.
Any other questions? Do you think or do you envisage that with some of these advances we can get to a point where we could actually prevent diabetes completely? That's a little bit of a loaded question, but I think I can't resist that one.
Yeah, I mean, it's a very good question, but as a regulator, you know this better than I do, that for the disease like type 1 diabetes, where you have insulin, right? You don't want to take the risk of treating an individual who does not yet have the disease with an experimental drug, so to speak, that has not been vetted through the clinical testing process, right? Eventually, if these drugs are proven to be to operate in this with the same efficacy as they do in animal models, and we if they perform in the clinic as expected, then yes, by all means. I mean, there is a possibility that one might intervene before patients develop hyperglycemia if we have indicators of ongoing beta cell autoimmunity, like presence of autoantibodies and so on and so forth.
Yeah,
The interesting thing is that I'm sort of looking at, and I'm not looking at this really, I kind of take off my hat sometimes as a regulator. I have my hat as a regulator, but I also have my hat as a physician, and my training had been in endocrinology, so I kind of, this is an area that I kind of have a fondness for. And in particular, I think, diabetes, it's the issue, as you put it, is very critical, because this is a bit like a vaccine. You want this to be really safe, so that if you prevent the disease, it is prevented and there's less toxicity, and it should be easier to use, in fact, than the insulin, for instance. And over a long lifetime, it should not provoke any major issues. And that usually is a long process, and that's quite clear. But I think on a theoretical basis, could one conceive that some of these immune diseases could be actually cured with some of these platforms? In a sense that we are having, even in rheumatoid arthritis, which is kind of treated at a reasonable early onset, you would then treat them for a number of years and it would not recur, or maybe retreat them if it recurs, but you wouldn't have to have continuous therapy.
Right.
You think that there is a potential in your platform?
Yes.
At least on a theoretical basis.
Yeah, no, absolutely. All the mechanistic experimental data in animals suggests that to be the case. I mean, it's an active, our therapeutic approach involves an active process. So we're not, and like the case when you use, monoclonal antibodies that block cytokines or receptors, it's sort of a passive therapeutic approach. You're blocking something that the immune system has because it needs to operate. In our case, we're inducing something that does not exist at the numbers that you need for the disease to not occur, and then these active processes is long lasting. Now we do not know how long lasting will be in patients, and that's why we need to do clinical trials. But we will certainly find out, and that's the hope that as we expect based on the animal data, that these drugs will induce reversal of autoimmunity without the need of continuous treatment.
Fortunately or unfortunately, I was marginally involved in the original cyclosporine trials, so it's an area that has been, I think, I've been fond of ever since.
No, thank you. Very good questions.
Yeah, maybe I can add something to.
Any other question from anybody?
I could maybe comment on just in addition to what Per said, because he made the comment that, you obviously with a prevention type of therapy, you want to be safe, or you want to be very sure that that person is going to go on to develop diabetes if you're going to intervene. Diane had spoken to, the power of autoantibodies in predicting disease, and that's the best we have. But even, you have to sometimes go out, 10 years, 15 years for double antibody positive people to turn diabetic. So I think that's why, we're so interested in multiple biomarkers and further research into trying to increase our power to predict disease more precisely.
Thank you very much. An area to really watch for for everybody, I think, carefully, because it holds promise. And we hope that the promise is going to be fulfilled. Lots of work left to be done. I just want to bring to your attention, please, that there if you look at the chat line, there is a little note both in English and in French, and a link to to forms and to an assessment of the conference. If all the attendees could please try and do their best and answer. Thank you very much.
Dr. Klein, there is a question in the chat.
Pardon me?
There is a question in the chat.
There's a question in the chat.
Yeah,
that
I can read it out if there's a comment and a question.
Okay, thank you, please.
So the question's from Kathy Pettigrew, and it says, she asks, with beta cells not totally being destroyed, am I correct to understand that someday there may be a way to restore them to full function? And I guess that might be partly targeted to me, and where I was talking about residual beta cells. And we just had a panel talking about this at the NPOD meeting yesterday, and the short answer is yes, that there are drugs that we could potentially target those beta cells to wake them up to improve their function. One of the potential dangers there is that one thing we're not sure of is that these cells that are beta cells that are still around, whether they are escaping autoimmune attack for some reason, that they are not as mature, or they're sleeping in some way. Maybe they're not expressing some of the antigens that are attacked by the immune system. And so in waking them up and improving their function, that might make them more subject to attack. So that's a concern, but that will have to be, sorted out first in preclinical models. We don't really understand enough about those residual beta cells right now. It seems likely to me that a therapy, a non-prevention therapy, a therapy to reverse diabetes is, and maybe Dr. Santamaria's therapy will reverse, but it's also likely that we'll need both immune therapy and some therapy to restore the remaining beta cells to sort of a combination therapy.
Anyway, unfortunately, we're out of time. This has been really fascinating, and the questions that were posed were very good and extremely useful. Thank you so much. Can you please stay for a few minutes for a couple of closing remarks that have been actually pre-recorded due to a lack of availability of the people. Well, one of them is, but the other one is not. So, Andrew, can you put them up, please?
Good afternoon. It's my pleasure to be here to do the closing remarks to this remarkable session. I would like to first say a big thank you to all the distinguished speakers for sharing their knowledge and experience with us today. We spoke about a number of really important elements, from the management, treatment, and management of type 1 diabetes, all the way to the prevention of type 1 diabetes. JDRF's motto is from type 1 to type none, and that's why we got here together today in order to discuss how can we get to that vision of type none. I would like to also take this opportunity to say a big thank you to JDRF for allowing us to co-host this session today in order to delve into these important elements of the treatment and management of type 1 diabetes. These patients face all sorts of challenges on a daily basis, regardless of whether there is a pandemic or any other issue. And that's why we're here together today, is to discuss how can we make their lives better. I look forward to seeing more sessions like this in the future, and Health Canada is very much interested in continuing these conversations as research advances and treatments emerge, so that indeed we can turn type 1 into type none in the future. Thank you, and wishing you all a wonderful rest of your day. Thank you.
As bringing breakthroughs to life is coming to a close, I want to thank everyone that was involved in making today's event such a tremendous success. I want to thank Health Canada, our co-host, all of our attendees, the moderators and presenters, for giving of your time because it made today such a rich experience. You helped us deliver a symposium that created dialogue, knowledge exchange, and you certainly saw firsthand lots of new and emerging treatments for type 1 diabetes. And I'm convinced that with ongoing collaboration and partnership, we can ensure that people with type 1 diabetes will live healthier, easier, and safer lives. And ultimately, we will be able to give them freedom from this relentless disease. On behalf of everyone at JDRF and the type 1 diabetes community in Canada, thank you for joining us today.